So should I keep my video on or I should switch off my video?
Sir, it's up to you. If it's video is on, then it would be fine. So everybody can see you.
Okay. No.
Next week.
Dr. Bhanakumar.
Yeah, yes, sir. Yes, sir.
Introduce sir first up.
Yes, sir. I am
please
just
introduce,
waiting for a significant amount of participants. By
Okay, okay,
9:5 we will start, sir.
Okay.
Yeah, sir. Yeah, that's a great topic. Sir, yesterday you referred to MD. Actually, I am really sorry that I don't know the terminology. Anyhow, I thought it's molecular dynamics.
Yes.
But it's molecular docking.
No. MD is molecular dynamics also.
Okay.
Okay. So I asked actually about the molecular dynamics because these will followed by molecular dynamics.
Okay. Okay, then we will start this session. I think Madam will join in a few minutes. So later on we can introduce her. Right now, I welcome all the participants to this FDP Day 2. Today there will be 3 sessions. Maybe fourth session also will be there. And on behalf of our director, Professor Dr. Pinkeshwar Mahanta sir, our institute, and our participants, including our chair for today's session, Dr. UK Shah sir and Dr. Manjula Gatak madam, and myself, I welcome today's expert, Professor Dr. Parimal Kar. Sir is a assistant professor at IIT Indore. So I will read out the achievements of sir. So which is quite I don't know the word. It is tremendous. Okay, so sir, I would like you to unmute the display, and I just want you to come on video, sir.
Okay.
So that our participants can see you. Yeah, sir. Good morning once again. So, sir has done his undergraduation from University of Kolkata, graduation from Indian Institute of Technology, Bombay, and PhD from Michigan Technological University, USA. So he has done his undergraduation and post-graduation in physics, and continued his PhD in computational biophysics. including physics at Michigan Technological University, USA. He has a lot of work experience, starting as a visiting student, then visiting student at Germany, graduate intern at USA Los Alamos National Laboratory, visiting scholar again at Germany, guest scientist at Max Planck Institute of Colloids and Interfaces, Potsdam, Germany, 2010 to 2012. Visiting research associate, Michigan State University, 2012 to 2016. Then he is now Ramalingasamy Fellow at IIT Indore since 2016. He has received a lot of he has received a lot of awards and fellowships, starting from student fellowship Germany for 2006, 2007, 2008, Biotechnology Research Center Travel Grant Award from Michigan Technological University 2007, IGRTG Postdoctoral Fellowship German Research Foundation 2011, Postdoctoral Independent Career Potential Award, Michigan State University, East Lansing, MI USA 2014. I think MI stands for Michigan State, I think. Ramalingaswamy Fellowship from Department of Biotechnology, India, 2015. So he has been a member of American Chemical Society and Biophysical Society. So his research area includes development and applications of coarse-grained and multiscale all-atom coarse-grained methods for biomolecules and the mechanistic study of large biomolecular systems, multiscale modeling of biomolecular recognition, protein ligand interactions, protein membrane interactions, free energy simulations and enhanced sampling methods, modeling host pathogen interactions in infectious disease and innate immune response, interaction of carbohydrates, glycans with proteins and membrane. His publications, there are tremendous amount of publications. So I am lost at those publications. He is having publications with in PLOS, then Journal of Biology and Chemistry, if I am not wrong, Advances in Protein Chemistry and Structural Biology, then Journal of Chemical Theory and Computation, again Journal of Chemical Theory and Computation, then Journal of Physical Chemistry, then Transcriptase, Journal Physical Chemistry B. Yeah, there are a lot many journals from Sir, and his research is being carried out at his own lab. So his broad areas are computational biophysics. There he has a lot of research scholars working under him. So now it's over to our expert. And I once again remind our participants that we are joined by two of our faculty members. So now I will hand over this session to our expert, Professor Dr. Parimal Kar. Sir, please.
Thank you, Dr. Vanav, for such an elaborated introduction. Okay, thank you very much. And also, I would also like to thank NIT Arunachal Pradesh for giving me an opportunity to interact with you all. So, I came to know from Dr. Vanav that the participants are with various backgrounds, actually, from physics, chemistry, engineering. So therefore, I choose this topic, molecular talking, because it is an interdisciplinary topic. And this topic is very pertinent to the current scenario, because you know that due to this ongoing COVID-19 pandemic situation, the world is really required a drug on urgent basis. Having vaccine is good, but that's not the solution actually. You know, vaccine is a preventive thing. But on the other hand, if someone is infected, you want to cure that person. So for that one, you need to have a drug. And over the last couple of months, I mean, since the outbreak of COVID, although the drug is still elusive, but we have thousands of papers that talks about that they have found the magic molecule. Okay, but that's not the case. So now you know that people talk about the remdesivir is the magic molecule, but you know that that's not the case. Remdesivir was designed for Ebola. Okay, so that's not for COVID. So the question is, the whole world is working to develop a drug. Last started maybe from January. This January we started, and it's almost October, and still we don't have the drug. So why is it so difficult to get a drug? And what are the bottlenecks? How generally the drug is developed? And as a computational scientist, what would be your contribution to this drug discovery? So we'll mainly talk about those things. Okay, different algorithms that is used in computational techniques to design a drug. And what are the bottlenecks? What could be the future directions? And also I'll show you some examples of our work on COVID-19 drug discovery. Okay, so the first question is, why does it take so long time to develop a drug? So these 10 months have passed, we don't have the drug. And 10 months is very, very less amount of time. Generally, it takes 10 to 12 years. Actually, 12 to 15 years previously it used to take, 10 years back, to develop a drug. And it costs you more than billions of dollars. Then finally you get the drug. So why does it take time? Because from the project team and these plans, so when they say it, okay, they talk about the plan, okay, this is the disease and this is the target, so we need to develop a drug targeting this particular protein, okay? So then they come out with different compounds, they synthesize those compounds, and they analyze, and then they got the drug discovery, okay? And then once they got the molecule, then you need to do the safety training, the early safety studies, that you do it on mouse model. And actually after the mouse, you go to monkey model, because monkey is more related to us, human beings, okay? So you check whether the molecule that you have developed is whether it is toxic or not. So if it passes, then you generate extensive, basically then you go ahead and develop the candidate, and then you go for extensive safety studies, okay? So large amount of candidate medicines are synthesized. Now, most of the cases, the molecule that we design actually fails in the very first study, early safety study, that we find that it is toxic. If it is toxic, we find, then again we go back to the first step. Again we synthesize a new molecule, again go for this safety study. So for example, this safety study you pass. Then you go for the phase 1 clinical trial. In phase 1 clinical trial, you study this thing in healthy volunteers. So those who have no disease, those who are healthy, you want to study on them and see whether there is any effect or not, whether any deterioration or not. If it fails there, highly chances it's going to fail there. Then again you go back to the stage 1. Again, start again doing from the very beginning. Suppose you pass the clinical stage 1, then you go to the clinical stage 2. In clinical stage 2, you have 100 to 300 patients. So those who are really suffering from the disease. So now you are really applying onto the infected part. So if it cures them, or there is no side effect, then it is fine. But if it does not, then again you go back to the first step. If you pass clinical stage 2, then you go to clinical stage 3. And there you have 3,000 to 10,000 patients. And then you test it on them. The data, analyze that. If it passes, you go for registration. And then finally, production. Okay? And if it does not, then again you go back to the stage 1. The problem is that there is a high chances that you will fail in each and every step. In many cases, it has been observed that you fail at the 3rd clinical stage. But from the conceit to the project and to the clinical stage 3, already you have spent 8 to 9 years. So if a project fails after 8 to 9 years, so you can imagine how much money you are incurring. So that's why actually drugs are also costly. Because when you get a successful drug, you don't know how many years have been spent and how many times they have failed actually in designing the drugs. So now in whole this scenario, where do a computational biologist fit? Okay, it's not only computational biologist, any computer, I mean, it could be a computer scientist also, because this field is basically amalgamation of computational physics, chemistry, computer science, and biology. All these 4 people come together. Okay, if you know only one field, is not good enough to work on this area. You have to be good in one area, but then you should have good knowledge about the other areas also. So the idea of computational scientist here is how can you minimize the time? So experimentally, when you want to synthesize large number of molecules, it's a very time-consuming thing. Go to any synthetic chemist, ask them, I mean, to synthesize all these small molecules. It's not that easy actually. It's a very difficult task. So if you ask them, okay, why don't you synthesize 100 different small molecules with which you will work on? So it will take them a lot of time. Not only a lot of time, it will cost them more money also. When you go to the wet lab, okay, when you enter into a wet lab, actually you are incurring money. It costs a lot of money. Okay. So as a computational scientist, our job is how can we minimize that number of starting molecules with which experimentalists can work on. Okay, and also we want to give them certainty that it's a highly possibility that these 5 selected molecules out of 100, I'm saying that, okay, I'll give you 5 molecules, and there's a high chance that of these 5, maybe 1 or 2 molecules will work. Okay, so that is our job. And to achieve that goal, we use this molecular docking techniques. Okay, so this is called computer-aided drug designing. So it has different component. Okay, so first one is the database. So what is database? Database is basically a small molecule database. Okay, where you have the 3D structure, or it could be molecular fragment database. So it's basically millions of compounds are there. Not only millions. Recently we did basically screen billions of compounds. Okay, so now the question is, now how does a computer scientist fit here? Well, you have to screen billions of compounds, but within a very short time span. How to do that? You need to bring in artificial intelligence or machine learning approach so that you can effectively screen billions of compounds within a reasonable time frame. If your database is large enough, there is a high chance that you can find a good candidate molecule. For a small data set, it's very difficult actually. Anyway, so once that thing is done, then other component is modeling. The problem is that you want to, for example, SARS-CoV-2, this COVID-19, okay? So you know that it is caused by this virus. You want to develop a drug, but you don't know the structure of the target. You want to target a particular protein, but you don't know the target. So although this disease broke out sometimes in November, December, as per China, it has broken out in December, but there is a high possibility that actually last year, August, it broke out. But the fast crystal structure of the target protein available sometimes in February or March. So until unless you have the structure, you cannot develop a drug, because whom do you want to target? You don't know. So in that case, how can computational technique help you? Well, we can model the structure. If we don't know the three-dimensional structure of the protein, we can model it based on the sequence. And that thing is called molecular homology modeling. Okay. We may not know the crystal structure of it. Right now we know it, but initial times we may not know the crystal structure of the main proteins of SARS-CoV-2, but we already know the crystal structure of the main proteins of SARS-CoV. That is the first SARS that was there in circa 2003, I think, that broke out in China. Okay, so that structure is available, and the current one, SARS-CoV-2 and SARS-CoV, has more than 90% similarity, sequence similarity. Only a couple of places there is differences. So of course, then you can build your structure based on the available older structure. Okay. So this is called homology modeling. So you found the homologs and then you build the structure based on that. Okay, so that is the thing. Now, once you have the database and you have the target protein, then your job is to do the screening. You have to screen the database, all the molecules that you have in your database, you screen it. Screening means to find the best candidate which binds very strongly to the target. Okay, and this screening could be structure based, shape based, or pharmacophore based. We'll talk about this in sort of different terms. Okay, and main thing about competitive drug design is first you need to identify the target. Which protein you want to target, which signaling pathway you want to target. That you can get it from bioinformatics analysis, or you can get it from the experiment that which proteins are playing a main role in the replication cycle, viral replication cycle, or any other important biological activity, so that you want to inhibit. Okay, and then you do the target validation, then you do the docking, scope, and you design the library. So once you get that thing, then you do the optimization of the molecules. Okay, that is based on the quantitative structure activity relationship. Okay, so finally, once you optimize it, you will have the drug candidate, and then you do the affinity evaluation. That means how strongly it binds. Okay, and also the ADME estimation. Okay, so how quickly it absorbs, what is the half-life of the drug, and what is the toxicity level. So that all those things you will study. So after you ascertain all those things, then you can tell your experimental collaborator that, okay, in my opinion, these are the 2-3 molecules that you can synthesize and work on it. So now you see that you started from millions or billions of compounds, and then finally you are suggesting your experimental team 5 to 10 molecules that they should work on, because you already computationally studying the toxicity level and all those things you are studying. So there is a high probability that in experiment also this thing will be replicated. Okay. Now, of course, it may not. Ultimately, computational is also just a modeling. Modeling means you are making some approximations. Okay. So all models are wrong. Why wrong? Because you are making approximations. But some of them are useful. That you should remember. Okay. We make models making some approximations. Some of them are useful, some of them are not. And generally, we make approximation based on what problems we want to tackle. Okay, so often this in experimental study, the computationally designed molecule fails because our approximations are wrong. Okay, so we need to go back to the basic physics and make correct approximations, and again revalidate or rescore and predict new molecules that can work. Okay. But definitely it is better than working 5 to 10 molecules. It's far better than working with more than 100 molecules. Okay, so that way you can aid experimentalists to reduce the time and thereby basically the overall cost you are reducing and also the time frame also. So these days we can get drug actually in 3 to 5 years. 7 to 8 years back, it takes 12 to 15 years. Now, with the advent of high performance computational power, especially the graphical processor unit, that is the GPU, that mostly kids are using it for gaming purpose. So that graphics card can be utilized to speed up your simulation. And thereby you can reduce the time, actually, overall time frame in the drug discovery. Okay. So now, what are the different two components of the drug discovery? Generally, this is called structure-based drug design, and other is the ligand-based drug design. Okay. So when you know the structure of the protein, okay, that is your target. When you know the structure of the target, then you go for structure-based drug design. Okay, so you know the structure. Now you have the database of ligand and you screen it and try to find out which molecule binds very strongly to the target protein. Okay, and if you don't know the ligand, then you have to de novo design it. Okay, that is another thing. Now, when you don't know the protein structure, then you have to do the ligand-based drug designing. Okay. Generally, you go for similarity searching and pharmacophore searching, and then this quantitative structure activity relationship. Okay. So, and if you don't know the protein structure and also you don't know the ligand structure, then computer-aided drug design will be of no use. You have to know at least one of them. Either you know the ligand or you know the target. Either of these you need to know. Better is if you know them both. Then it's the certainty, the accuracy level would be much higher. Okay, but generally, when you want to target membrane protein, for which it is very difficult to get experimentally determined crystal structure, you have to actually go for this ligand-based drug design. It is very difficult to get a membrane protein structure. It is very easier to, well, it's not easy, but comparatively easier than membrane proteins to get a crystal structure of the soluble protein. Okay, membrane proteins are hard to crystallize, actually. So for that purposes, mainly we use ligand-based drug designing, and for globular protein, also soluble protein, we use basically those proteins which are situated in the water environment. Okay, so those are called soluble proteins, for which we can use structure-based drug design because we know there is the structure. Okay, so these are the different steps that we use for structure-based drug designing. First is the target identification and selection. Second is once you identify, then you have to isolate it and purify. and then get the crystal structure, okay, via X-ray crystallography or NMR study. That you have to get it. But generally, if the protein structure is relatively large, then NMR may not be suitable. So we use X-ray crystallography. But these days, people use cryo-EM microscopy to generate it. India has one cryo-EM. Okay, so that is installed in NCBS Bangalore. Okay, it's cost around 50 crore. So that's a national facility. So the last year, actually, Nobel Prize went to for this cryo-EM, discovery of cryo-EM microscopy imaging techniques. So once you determine the structure, then you analyze the structure for potential ligand binding site. Okay. So once you have the structure, you find a pocket, because you have a small molecule, you want to bind it. So where it can bind? It can bind in a pocket. Okay. So you need to find out the pocket which can accommodate this ligand. And then once you identify the pocket, then you dock it. Dock it means you put the ligand in the binding pocket in a pose, okay? That means in a orientation, so that the interaction between the target and the drug is maximized, but the overall energy is minimized. Why? The lower the energy, the stable the structure is. Okay, you know that. I mean, if your potential energy is minimum, then only it's stable conditions. Okay, your stable condition will be when you lie on the bed, because your potential energy is the minimum at the time. If you stand, it is not a minimum energy. Okay, so of course, then lying down on the bed is a more relaxed position than standing actually. So same thing for protein drug interactions also. You need to maximize the interaction between the protein and the ligand. At the same time, the overall energy, we call it the Gibbs energy. Okay, that should be minimum. That should be more negative. Okay, the positive means they are not binding. It is not a stable structure. They will move out. Okay, so that's the thing. Once you up to this, you so this is computational study. Then you go for the experimental. Okay, you make biochemical assays and do the further testing, and then you go for lead optimization. Okay, for improving the potency. That means once you study these protein drug interactions, then you know that, okay, maybe if I substitute this particular functional group of the ligand by this particular functional group, then the interaction would be maximized. If the interaction between the protein and ligand is maximized, then they will bind tightly. That's your job. You don't want a drug which weakly binds to the target. The tighter binding can be obtained by maximizing the interactions between them. Okay, so first you have a molecule, then you optimize that molecule. You delete some portion and add some portions to it. Okay, which makes chemically makes sense actually. You should not design something that cannot be synthesized. Okay, so then you try to maximize that interactions, and then finally you will have your drug. So still it's a candidate molecule. Then you have to go for testing cytotoxicity. Because now you have deleted some portions and add something into it. So of course now you need to go for the cytotoxicity testing. If it passes that, then only it could be a drug candidate. Okay, then you should go for clinical trial. So these things take overall, I mean, it looks very easy, but it takes actually more than 2-3 years. Easily it takes. So that's why drug discovery is a very long process. So when we saw that it's already 10 months have passed, why there is no drug? What the hell the scientists are doing? Well, I mean, it's not that easy. Although all the methods are in place and many of the experiments have been robotized, but still it is not that easy actually. Even if your cytotoxicity, all these things are passed in animal models, okay, in mouse or monkey models, there is a high probability that it will fail in the human model when you really apply it. And sometimes you know that, so right now the situation is that they want to make avail the vaccine by next year. So how is it possible within 1 year? So what they are studying that, of course they are doing human studies and all those things they're doing. But here the main goal is not that they're getting the approved level of efficiency. So when Ebola broke out, okay, that was in African country, so they also developed the vaccine very quickly, and they started administering it. Who gave the permission? Okay, you just administer it. Why? Although there is no proof that it's going to work, but one thing was sure, that it does not cause any side effect. So if it does not cause any side effect, you can let it administer on humans. So at least if it does not do any good, at least it will not do any harm. So currently the situation is like that. If it cannot do the good, at least it will not do the harm. If you make sure that, then of course you release it. But when you really want to have a good vaccine or drug, then you have to make sure that actually it works. Okay, it prevents. Currently it's not like that. Nobody is sure. They don't have enough data set because we are in a hurry. The world is demanding, okay, we need something. Okay, you get it. So you take it, but whether it will work or not, I don't think that everybody is so much sure about it. One thing, just we need to make sure that there is no side effect. Currently, that is the strategy. And also, all these big pharmaceutical companies, they have invested a lot of money for COVID-19, and they want to release the drug as soon as possible because they know that in India, at least in next 3 months, 40 to 50% people will be infected. Whether you will know or doesn't know, that much percent people will be infected. It's just that you won't have any symptoms, so you think that you are not infected actually. But if they do the serosurvey, then it will be known that already 40 to 50% people are infected. Then what's the point of having a vaccine? If already that many people are infected and they have their own antibody, so you don't need. But already they have spent the money, so they want to release the vaccine as soon as possible into the market so that they can make money. At least they should be able to garner the money that they already have spent for developing this vaccine. Okay, and this drug will, of course, this virus will, of course, have a natural death. I mean, it is right now pandemic, but by next year summer it will be endemic. So that's why the companies are also in a very hurry. Otherwise, it is not possible to get a drug or a vaccine in 1 year. Forget it. It's not that easy. It takes at least 3 years minimum. It should take. Anyway, so now that is a structure-based drug design, and then there is a ligand-based. Okay, so it relies on the knowledge of known molecules that bind to the biological target. Okay, and this is called the structure and bioactivity. That is called IC50 value. And these molecules may be used to derive a pharmacophore model, which defines it's an abstract concept basically, okay? So that defines the minimum necessary structural characteristic a molecule must possess in order to bind to the target. And the main thing that is here is the virtual screening, okay, based on the pharmacophore models, including dark property filtering. You can use the Gene database, so that is a free database that you can use. And there is another KMBL database also that has also millions of compounds, that is also free. And alternatively, you can use the QSAR approach. But one thing I just want to tell you that in the all these the database that you have, all the molecules may not be available in the market. If it is not available in the market, you have to synthesize it for the experimental study. Of course, not for the composition study, but for experimental studies. But many of the molecules actually you can buy from the market. So once you can identify one molecule, you need to check it that whether it is available in the market or not. Okay, so then you can buy it from the market and then you can do the experimental study. So I don't want to talk about it. Okay, so next thing, this is the very important concept in computer drug design, is the virtual screening. So this is the 5 step of molecular talking. So virtual screening is the computational or in silico analog of biological screening. Okay, experimental thing basically. So here the aim is to score, rank or filter a set of structures using one or more computational procedures. So that means you have millions or billions of compounds. You want to screen it. Means you want to calculate their binding energy. So binding energy determines the strength of their binding. Okay, for all these molecules you compute it, their binding energy, and rank them based on their binding energy. Then you select the top 10 molecules or whatever you can. Okay, based on that. So that is their job. It's a very fast method. If it is not fast, then you cannot screen millions or billions of compounds. Okay. So this helps you to decide which compounds to screen experimentally. Okay. Because from millions to compound, you have to tell maybe 10 or 20 compounds that can be screened experimentally. Okay. And then which library to synthesize. Okay. Then you say, okay, only these 20 compounds you synthesize and go for the testing. And which compounds to purchase from an external company, if it is available in the market, of course, and to analyze the result of an experiment such as high throughput screening run. So that is the job. So virtual screening is a very fast method. And in computational technique, remember one thing. You always need to make a balance between speed and accuracy. If you want to give a preference to speed, then you have to forget the accuracy. And if you want to give a preference to accuracy, then you have to forget about the speed. So we need to make a strategy that can make a compromise between speed and accuracy. We want to take a middle ground. Okay? The method should not be too fast and it should not be less accurate either. Both should be in a moderate manner. So that's why we adopt multi-stage approach. First, we do this virtual screening. From virtual screening, we found thousands of compounds. And from these thousands of compounds, I use rigorous method to generate 20 molecules. From these 20 molecules, I even use another set of more rigorous method and finally come up with 5 molecules. So you can use this multi-stage approach, and in each stage you are using more accurate method, but at the same time the speed is less. But since you are using this more accurate method on a small subset, so you can definitely afford it. But in this whole process, you should have a HPC system, okay, high performance computing systems, basically. So the virtual screening, basically, it acts as a, you can have some general filters, okay, of course. When you reduce the number from millions to thousands, so of course you have to use some filters, okay. So the substructure filters, it could be dark bright filters or the AD-emissive properties, okay. And then if the 3D structure known, then you will process structure-based method. Then you will do the docking study. Okay, after that. And finally, you will do the biological screening, compound acquisition, library design, and synthesis, and go for the clinical trial. So that is the whole thing. Your job is up to put in structure-based methods, basically docking, and tell the experimentalists these are the 5 compounds. Your jobs end there. But of course, theoretically, you can predict all these biological activity also you can predict. And that can be assessed in experiment also. Okay. So the main thing is now molecular docking. Okay, because virtual screening, you use it to reduce the number from millions to thousands, and now you use molecular docking to reduce the number even further. Okay, so docking is relatively a better method than virtual screening. Okay, it's more accurate, but it takes a little bit more time compared to the virtual screening. So before that, we should know what is molecular reconnaissance, because protein-drug interactions means how the target molecule is recognized. It's recognizing the ligand, that is your drug. You need to know that, the process. If you know, then you can modify your drug candidate and make it a better drug. Okay? So that's the lead optimization. Basically, you can optimize your molecule and make it a better candidate. So that means you need to know this recognition phenomena, how these 2 molecules are recognizing each other. When you take a drug, so basically it's a free molecule, it's coming to your body, and the protein is also free. Both these protein and ligands and proteins and the ligands are free molecules. They are rotating, vibrating, translating in your cell. So how they are recognizing each other and binding. Okay, so you need to know that thing. So that is called the molecular recognition. Okay, so molecular recognition refers to interactions between 2 or more molecules through non-covalent interactions, including hydrogen bonding, electrostatic interactions, and hydrophobic interactions. Okay, so some of the amino acids that protein compose, actually those are hydrophobic. They don't want to interact with water. Okay, they are buried. Since they are buried, actually that makes your protein structure compact. Now, otherwise it will be bulky. If it opens up, your structure will be bulky and it may fall apart. Okay. And hydrogen bonding is also electrostatic. Of course, it is electrostatic. It is ionic interactions. So it is also very important because it's strong interactions actually. Okay. So if your ligand actually forms hydrogen bond with the receptor, that is the target, then you will have a strong binding. But that was the main conviction. In the drug industry, people actually they used to think that if we can somehow increase the number of hydrogen bond between the protein and ligand, we will have a very strong binding, and that would be excellent drug candidate. But that's not the case. They have studied actually that they increased the number of hydrogen bond, and finally what they got it actually they even got a worse candidate. The binding strength has been reduced. Okay, the question is why? That's very important actually. In experiment that you cannot tell actually why it is happening. So as a computational scientist, you can tell actually what is happening. Why forming hydrogen bond means actually more stronger interactions. But what you were saying that if you have more number of hydrogen bonds, actually you are not getting desired strength, increase in strength. It remains same or actually decreases. Why? Okay, for that one, we'll talk about it over the time. Okay, so all those questions can be answered by computer simulation studies. Okay, so molecular recognitions. So basically, all these different interactions we study, and it is very important because it helps, molecular recognition actually that promotes the fundamental biological events such as transcription, translation, signal transduction, transport, regulation, enzymatic catalysis, viral and bacterial infection, and immune response, all those things. But you are not interested in those things, okay? Although all these biological phenomena basically boils down to the protein drug, or protein sugar interactions, okay? What actually you think that cell-cell interactions, actually it may boils down to protein glycan, that is the sugar molecule interactions in many instances actually. Even for this SARS-CoV, this virus, okay, so you know that this virus glycoprotein actually, it attaches with the human ACE2 receptor, okay? Now, so this whole infection process, okay, now this glycan of this, this glycoprotein of the virus, on their cell surface, they are nicely decorated with sugar molecule. So basically, the human ACE2 receptor recognize this sugar molecule that is decorated on the surface of the glycoprotein of virus. So you see that at the very molecular level, this infection process is nothing but interaction between protein and the sugar molecule. So that is also a recognition event. How your protein, that is our human ACE2, is recognizing the sugar molecules of the viral glycoprotein. Okay? So basically, virus makes sure that our human ACE2 protein thinks that the glycans that is situated on the viral glycoprotein surface is self. So it cannot differentiate between self and non-self. Our ACE2 protein thinks, oh, no, these glycans is actually, maybe it's from the self, it's mine actually. So it binds. If it thinks that it is foreign, so it will not bind, but it cannot differentiate. So that's why it binds. Once it binds with the viral, these glycans, that's a sugar molecule on the surface, then you got infected. Okay. So now if you can target that interaction, that would be great. That way you can develop a drug. Now to develop a drug, you need to understand this recognition event, how they are recognizing each other. Then only you can inhibit this recognition process. Okay, so that is the importance of starting the molecular recognition process. Okay, but we'll not go into this host pathogen interaction thing. That is beyond the scope of this lecture. So today we'll talk about this drug target protein interactions. Okay, so the main thing is that how the proteins look at the foreign molecule. That you need to know. If you want to design a drug, you need to understand how the protein sees the drug. The chemist sees the drug aspirin and sildenafil. Sildenafil is the Viagra. That's the commercial name. And aspirin you all know. So this is how the chemists see. This double bond, this, that, single bond, nitrogen, oxygen, sulfur, all these things. The chemist sees like that. But how the protein sees it? The protein sees aspirin as like this. It sees as a surface. It's a van der Waals surface. And on which, on the surface, you have basically, since all the atoms have charges, point charges, okay, we are talking about the classical mechanics, okay, so in classical mechanics, each atom has a point charge, that is called Mulliken charge, you know that, okay, if it is quantum mechanics, there is no such charge, we have electron cloud, okay, we will not talk about that thing. Let us concentrate only on the classical aspect, okay, so each atom has a point charge, so that means there will be charge-charge interactions, electrostatic interactions, okay, that you can get it from Coulomb interactions. Remember one thing, the Coulomb interaction is pairwise interactions, okay, it is between two atoms. Now each atom can have so many number of pairs. It can form, one atom can form pair with the rest of the atoms, okay, n-1 atoms it can form. So each atom will have that kind of interaction. So you can see that for a computation actually it is very challenging because so many pairs you will have and you have to compute for each cases. Okay, so you then calculate it and project it on the surface. So the red are negative and the blue are positive and this gray color is the neutral. So you see that the protein looks at like that. So now I say that you need to dock it to a binding pocket. Now how the binding pocket looks like? You can do the same thing. You can calculate the electrostatic of the binding pocket residue and project it on the surface. So of course, now you know that if aspirin surface is more negative that you can see, so you know that you should place it to a cavity where the surface charge is electrostatic potential is positive. Okay, then only it will have a attractive interactions and they can bind more strongly. Okay, so you have to have that kind of thing. Protein sees it exactly like that way. So instead of thinking of single molecule, single bond, double bond, all those things, basically you finally need to see how the protein looks at it. Protein looks at like that. In this below panel, you see this hydrophilic area and the reds are the hydrophobic areas and green are the hydrophilic. Okay, this is the protein surface actually, the binding cavity, how it looks like. It could be hydrophobic surface or it could be hydrophilic. If it is hydrophilic surface, that means it wants to interact with water molecules. So you need to make sure that there in the cavity there are some water molecules there. So you need to design a drug that can form hydrogen bond with the receptor via water molecules. Okay, so there could be a bridging water molecules. So that way you can satisfy it. Okay, because the binding pocket, which is hydrophilic, it is fast reacting with the water molecules, and the water molecule is reacting with the ligand. Okay, so you are satisfying both things because you need to make sure that if the surface is hydrophilic, it should react with water molecules, and if the surface is hydrophobic, then there should not be any water molecules in it. So your molecule, the molecule that you are designing, should snugly fit into the binding pocket so that no water molecules slip into the binding pocket. Okay, so that kind of strategy, okay, intuition that you need to develop. And all those things you can do it computationally. You can generate all these structures that in the afternoon we will tell you how to generate all this kind of representation. So from there you can actually sense it, what kind of molecule you need to have. Okay, that will help you to the candidate molecule that you get from the virtual screening, you can modify it accordingly. Okay, so now this understanding this mechanism underlying recognition is of great importance for rational drug design. Why? Because of course, if you know these molecular recognitions, it can act as a control. You can block the recognitions. Okay, that means those are called inhibitors, antagonists, etc. Or you can increase or emulate the recognition. That is called, that small molecule will be called agonist or recombinant proteins, etc. Okay, so that is why it is very important to know these fundamental this recognition process. Now you want to develop a drug. Basically, you want to dock. I said that. Okay, docking means placing the molecule in the binding pocket. It is exactly that. So docking attempts to find the best matching between two molecules. Okay, so that the small molecule, your drug molecule is small actually. Okay, the target is big. So that it's snugly fitted into the binding pocket. Okay, just like mommy snugly hugs their kid. Okay, it should be like that. This is just a cartoon representation, but the more serious definition is that molecular docking is the process that involves placing molecules in approximate configuration to interact with the receptor and form a stable complex with an overall minimum energy. Now, what is configuration? Configuration is nothing but the Cartesian coordinates. That is the x, y, z coordinate, okay, of the molecule, each atom basically. So any protein or ligand, okay, they are composed of atoms. So each atom will have a position coordinate, okay, x, y, z coordinate. So that is called the configurations. So that is molecular docking, and it's a natural process which occurs within seconds in a cell. But in computer, it does not happen, okay? It takes more time. Actually, we cannot simulate any event in computer at the time scale of second. Why? Because molecular dynamics simulation, we start from femtosecond time scale. Now, from femtosecond to second time scale, it takes billions of iterations. That is very hard to. We simulate, we start from femtosecond and we achieve, I mean, so far I simulate generally up to 20 microsecond simulations. So still it's a huge time. You see that from 10 to the power minus 50 to 10 to the power minus 6. So that means you have to do 10 to the power 9 iterations. Okay, we'll talk when we talk about MD simulations, okay? So that maybe in the next lecture we'll talk about it. So in molecular modeling, the term molecular docking refers to the study of how two or more molecular structures fit together, okay? So here is the protein, okay? And the binding site that I have shown in a red circle, okay? And the ligand is green color. So basically when they associated, exactly they form this complex, okay? So you see that how this ligand is occupied the position in the cavity, okay? So basically in molecular docking, you predict the pose of the molecule in the binding site. Pose means the orientation. You need to optimize the orientation such a way that the interaction is maximized, okay? The binding affinity or a score representing the strength of the binding. Now, which one is the best pose? How do you determine? You determine it from the binding affinity. The stronger the binding affinity, better the pose is. Okay? So that's the thing. So aim of molecular docking is to achieve an optimized conformation and relative orientation between protein ligands such as delta G is minimized. Delta G is the Gibbs energy. Okay? In thermodynamics, we call it Gibbs energy. And Gibbs energy is delta G is equal to H minus Ts. Okay? H is the enthalpy, T is the absolute temperature, and S is the entropy. So now, in experiment, you don't calculate the entropy actually. Well, in ITC you get it, but you don't really compute what are all possible interactions it can have. In ITC, that is isothermal calorimetry experiment, you get the delta H, that is the enthalpy and the entropic part. But what are the different components of the enthalpy? That you don't get it. Okay? So if you don't get that thing, then how do you optimize the molecule? You cannot optimize it. But for molecular recognition study, this docking study, we can actually tell what are the components of different parts of the enthalpy so that we can optimize it. Docking is whether two molecules interact with each other, and if so, what is the orientation that maximizes the interaction with minimizing the total energy of the complex? I told you that. A complex structure would be stable when the total energy is minimum. The more the negative energy, the more stable structure it will be. Okay, so your goal is to make delta G more negative. Okay, so given a protein structure, predict the ligand bindings. That is your docking. That exactly we'll do in the tutorial. Okay, how to do that thing. we'll take an example of, because right now everybody is designing drug targeting SARS-CoV, so we'll also take an example for SARS-CoV and exactly do the talking. Okay. Hopefully, after you do that, you can do your own study and publish paper. So I already talked about this, why talking is useful. I don't want to talk about these things. Okay. So now, what are the different models that is used for molecular talking? So in 1890s, so there was this lock and key model, which was developed by Emil Fischer.
Sir.
Yes.
Sir, sorry for interruption. Sir, we can carry on this in the next session, sir.
Okay.
Yeah, sir. Shall we stop here?
Okay. And then when we will start then next?
Next will be in the afternoon, sir, 1:45.
Okay. No problem.
Yes, sir. Yes, sir. Because it's time. It's almost 1 hour 15 minutes. You are being giving the talk, sir. Next few minutes, the participants may be raising few questions.
Okay, that's fine.
So yes. It will be I request all the participants to raise their questions in the chat box, or they can unmute and request their doubt. Yeah, I request the participants. Yes, please.
So a lot of students have asked where to get the slide for studying. So I will share the slide. The Friday I will have last talk. So after my all sessions over, I will share my slide with Dr. Banav, okay, so that he can share it with all. One has asked the dipole moment of biological molecules. Okay, so of course, dipole moment is very important. But one thing I just want to tell you that to introduce these dipole moment terms, okay, generally we need to use better if we use polarizable force field for this one. Okay, so the polarizable force field can take care of this thing. But remember one thing, that polarizable force field is relatively very slow. As I tell you that in molecular modeling, you always need to make a trade-off between speed and accuracy. So of course you can introduce dipole moment. If the molecule has, of course, high dipole moment, you need to take into account, but then at what cost? How much you will gain? If you don't gain much, but you are taking into account, because you are using a very sophisticated force field to model that thing, it may not be useful that much. Okay. So even for SARS-CoV, I'm telling you that NSP10 and NSP16, okay, this non-structural protein, these two proteins has very high dipole moment. Now they form a dimer. Okay. So you want to study their dimerization process. Okay, how they are recognizing dimerization? They are recognizing each other, no? Because those are heterodimer basically, NSP16 and NSP10. So how they are recognizing each other? Both have very high dipole moment. Of course, they will play a role. Now you want to study that thing, influence of dipole moment, and you have to use polarizable force field, but it's very, very slow, and you may not gain much. Okay. So my suggestion would be, you make a judicious choice. If you wanna include it. If you have good computational resources, why not? But don't use it in the very beginning. Okay. First you reduce the number. Use virtual scanning, reduce the number. Then you use, for the virtual scanning, actually you should also use different stage manner that I said. You use a different sophisticated method at different stage and reduce the number. And finally, once you make, say, less number of candidates, say 10 candidates, then you can use polarizable force field to model all these things, because you can afford, because it's just for only small set of molecules. Of course, you can afford it. But for large set, I would not suggest to do that thing. Okay. What is Monte Carlo simulations? Well, we'll talk about it. We have not yet gone to Monte Carlo and molecular dynamics. Okay. Over this weekend, over this week, I'll talk about Monte Carlo and molecular dynamics. Monte Carlo is basically actually, it's you are introducing some probability in it, and whether actually, let us say like this way. So protein has a very rough free energy landscape, okay? So, and it has a very conical free energy landscape. So this conical, the last cone, okay, that is your globular structure. That is your correct three-dimensional structure. And in this process, you have multiple minima. Okay, so it's a glassy system. In physics, we call it glassy system. Okay, so here, so how do you overcome those barriers? Because there are multiple barriers. Now you can get stuck in one of those barriers and you may not be able to overcome that barrier. And you run it for a long time and you think that, okay, there is no change in structure. So that means that I have reached to the equilibrium. Actually not. Actually, you got stuck in one of those local minima. That structure resembles the three-dimensional structure, correct three-dimensional structure, but that's not the exact structure. Okay, so for that one, you can use, say, Monte Carlo approach. In Monte Carlo approach, what do you do? What you do basically, so each stage has a different energy. Okay, now I make sure that I will move to the next stage only when that's the next configuration will have a lower energy, because higher energy means it's a wrong structure. Okay, idea is always to go to the energy level down and down and down. Okay, finally this to that. So I will take a state which will have a lower energy than my current state, current configurations. Okay. Now, so whether we will take that step or not is determined by a particular weight function. Okay, that generally we use it a canonical weight function. Because so Monte Carlo has a formula for that one. For which case we will take it and for which case we will not take the move. If the condition, Monte Carlo condition is, I'll show you later, okay. If the Monte Carlo condition is satisfied, we will move to the next step. If it does not, we'll stay at there. Again, we'll run it and evaluate the energy and again try to evaluate whether I should move to the lower energy level or not. So these help you to overcome the barrier basically. I'll talk about it, don't worry. And MD simulation is also kind of thing where in MD simulation actually you solve the Newton's second law. Basically F is equal to mass time acceleration. That is the equation. It's the second order differential equation, okay? Because acceleration is d2r dt2. So m d2r dt2 is equal to force. So this is the second order differential equation. And remember one thing. In molecular dynamics simulation, you are not solving this single differential equations. You are solving 3n number of coupled second order differential equations, and n is the number of atoms. So if your system has 1 million atoms, say not that much, say 1 lakh atoms. So then basically you are solving 3 lakh this second order differential equation simultaneously. Okay? And you are basically, it's a time evolution of position. How the atoms are moving with time. In Monte Carlo, you are not getting the time evolution. Okay? But in MD simulation, you are getting the trajectory as a function of time. How each atom is moving with time. So that is the advantage of molecular dynamics simulations. Now you can have a hybrid method. I can combine molecular dynamics and Monte Carlo together because I want to get the best of both. Okay, I'll show that thing also and why it is useful. Probably I'll show you one example also.
Sir, one of our students,
Yeah.
Chiranjeevi Vishwadeep Das, want to know what is fragment-based drug design? What is fragment-based drug design?
So fragment-based means that, so actually maybe in the afternoon session I'll talk about it, okay? Because we have not gone that far. So
and
basically you have the molecule, no, you make it into different fragments in its rigid part. So first you take one rigid part and dock it into the cavity. Then in the next step, you take the other fragment and add it. So basically you are sequentially adding it. Instead of docking the entire thing, you make it into different fragments and you are adding it sequentially. That way you can do it. Okay? You add it to the position, like that you can do it. We'll talk about it. Okay? Don't worry.
Okay, another participant, Chiranjeevi Pasala, want to know which docking is the best. Which docking is the best among different dockings?
Again, in the afternoon we'll talk about it because I am not done actually. So docking, best docking is the one where you are taking into account the flexibility of the protein as well as the flexibility of the ligand, because both are flexible molecules. Okay, that is the best. But the question is, again, I am telling you, at what cost? If you want to take into account the flexibility, protein flexibility is very difficult to take. Ligand is small molecule, you can take it. That is the most of the docking software do. They treat ligand as a flexible molecule and protein as a rigid molecule. Okay? But proteins are not rigid, proteins are plastic. Okay? It has a plasticity, it is flexible. And mainly, the flexibility of the side chains in the binding pocket, you are mostly interested in that because that takes part in the binding process. So you need to take into account that effect. So the best method is one which takes into account flexibility of both the protein and the ligand. But generally, all the docking software, I mean the best, the commercially available docking software, they take a semi-flexible approach. Okay, so what they do that, let the ligand be flexible. But for the protein, the side chain flexibility is taken into account by some rotamer libraries are there. Okay, that is predetermined rotamer library. That is actually knowledge based. Okay, that you get from the protein data bank, and you study from that. But that exactly does not mimic the real biology. Okay, so the best one I would say that available to you is Schrodinger software. In Schrodinger, that's the guide, and they take it in a semi-flexible manner. Really, it's not really not the fully flexible manner. So what you should do that, you use this semi-flexible approach. Okay, let the ligand be flexible and the protein be semi-flexible. Okay, and you dock it. After the docking, you run molecular dynamics simulation. So when you run molecular dynamics simulation, then you are letting both the protein and the ligand flexible manner. You are not putting any restraint on them. Okay, so that way you can take into account. So it will, of course, you are relaxing the structure and the ligand will also adopt its structure. So that's why I'm telling you, whatever you are getting from the docking is not the correct thing. Many of the times you will see that the prediction that you get from the docking is wrong. Actually, if you run very long time scale MD simulation, the drugs actually come out. I will show you some example also, that kind of thing. Okay.
Okay. Another participant, Devasish Mohapatra, wants to know what are the algorithms used for docking?
So it is different docking algorithm. Actually, I have, since I was asked to stop, actually, I mean, I am gonna tell you what are the different you have algorithm generally. So these are the different algorithms that I take. For the ligand specifically, we take these.
Sir.
Yeah.
Sir, our session chair, sir, we will leave this question to next session. Okay, sir.
Actually, because next is I will do all these things. Okay, so then you will have these questions will be answered because I have all the slides. Okay.
Yeah.
Talk in the next session. I will tell all things. Okay, I'm coming sequentially. I'll of course tell all those things and also I'll share the slide. Okay, don't worry about it.
Okay.
Sir, okay. So, yeah.
Okay. So, thank you very much, sir. Vanav sir.
Sir, actually, sir, our
thank you.
Session chair, Manjula Gatak madam has also joined. She has immediately joined. I would, so Manjula Gatak madam is faculty member in Department of Mechanical Engineering. She has been in charge of Women's Cell of NIT Arunachal Pradesh. So I just request Manjula Gatak madam to come on the screen.
Hello, good morning, ma'am.
Good morning, everybody. Good morning,
yeah,
sir.
Good morning.
Thank you, Chief Coordinator, sir, and thank you, Dr. Parival Kaur, sir, for taking out your valuable time. And I have attended your session. Is so much like full of information. We have researched quite well. Thank you so much for sharing your knowledge with us and being among us. Thank you so much. Thank you, everybody.
Thank you. So then I'll see you in the afternoon.
Yes, sir. You can leave for this session. Okay, sir. Thank you once again on behalf of our participants. Thank you, sir.
Thank you.
Yeah. So, participants, so I will be there for next 5 minutes. Then we can break for the next session. And I thank our chair and participants, our chair, Mrs. Manjula Gatak madam and Dr. UK Saha sir. Thank you, sir. Thank you, ma'am. Yes, sir. Thank you for coming. Yeah, participants, if you have any questions, you can put up. Next session will be at 11 o'clock. You are requested to join well in advance.